Alright, so next we're going to talk about transcription. Alright, so before we get into the process of transcription, it's important that you re-familiarize yourself with the central dogma of biology. Alright, so the idea here is that the information contained within the genetic code, right, is going to be transcribed into RNA. In this case, messenger RNA. This RNA is then going to be translated into a polypeptide or a protein. Alright, so for all intents and purposes, alright, this remains true. Except for the fact that DNA can be transcribed into RNA, and you have RNA that functions as messenger RNA, that functions as transfer RNA, that functions as ribosomal RNA, that functions as like regulatory RNA sequences, regulatory RNA molecules, like microRNA. Alright, so it's not always DNA, RNA, protein, because then you also have RNA molecules that can function as enzymes. Alright, so you have things called ribozymes. Alright, so ribozymes. Alright, which are RNA molecules that function as enzymes. Alright, so in this case, not all of your enzymes are proteins. You have RNA molecules that function as enzymes as well. Alright, so looking at this process of transcription and translation between your bacterial cells and your eukaryotic cells. Alright, so your bacterial cells, alright, you have your DNA, alright, you have your genes, they get transcribed into messenger RNA, and this messenger RNA is then going to make its way towards a ribosome, where it'll also interact with transfer RNA, and you'll form your protein, your polypeptide, right, where your RNA is translated into proteins. Now, notice, all of this takes place inside of the cytoplasm. Now, eukaryotic cells, the process of transcription is completely separated in terms of location from translation. Alright, transcription takes place inside of the nucleus. Alright, inside the nuclear envelope. Notice that also you have RNA processing. Alright, so you get your primary, alright, this pre-messenger RNA molecule is your primary transcript. Alright. That primary transcript has to be edited. It has to be processed before it can leave the nucleus. Because it'll leave the nucleus, it's directed to the ribosomes, where that messenger RNA is translated into a protein. Alright, so transcription occurs in the nucleus, translation occurs in the cytoplasm. You also have this editing process that takes place in eukaryotic cells that doesn't take place in prokaryotic cells. Alright, so going back to this idea of genes and proteins. Alright, for the longest time it was thought that there was, for every single protein, you had a single gene that was involved in making that one protein, making that one enzyme. So you have this idea of a one gene, one enzyme hypothesis. Now, for most purposes, that holds true. All right, and that most of your enzymes are proteins, but not all of them, because you do have RNA molecules that function as enzymes. But then again, also, not all of your proteins are enzymes. You have proteins that have many different roles. You have proteins that are involved in immune defense. You have proteins that are involved in communication. You have proteins that are involved in making up structure or structural components of a cell. You have proteins that are involved in functioning as hormones. And I think it's even more complicated. And that the idea was you have one gene, one polypeptide. Well, here's the problem. You can have one gene, one polypeptide, and that basically you have, for instance, thing like hemoglobin. Alright, hemoglobin is a protein that's made up of subunits. Alright, so it's made up of individual polypeptides. Alright, so you have an alpha subunit and a beta subunit, which have their own genes that control their synthesis. Alright, so you have two different genes, two different polypeptides. Alright, well that holds true. But you also have an instance where you can have a single gene that can produce many different products. Alright, so recall that there's this editing mechanism, right, that occurs in eukaryotic cells. So you can have one gene that could potentially produce many versions of a protein, depending on how it's edited. Alright, so you can get version 1 of that protein, you can get version 2, you can get version 3, all depending on how it's edited. Alright, so the sequence of DNA nucleotides serve as the instruction manual for the cell. Alright, so you have to convert this nucleotide sequence into an amino acid sequence. Alright, ultimately, that's the goal. To take the information that's encoded in the DNA and make a functional product, but it's protein. So it's made up of amino acids. Alright, so how exactly does this take place? Well, yeah, this process of transcription. Well, transcription is like rewriting the genetic code from DNA into RNA. Alright, so it's basically like you have, your notes. Alright, so you have student, for instance here, you have student A, and let's say that you borrow student A's notes, and you rewrite them. So now they're your notes. Alright, so you're rewriting it. That's all you're doing. You're transcribing. Going from DNA to RNA. Alright. And so let's say that this is all in English. Okay? Because, think about it. DNA, RNA. It's almost the same. It's a little different, but it's almost the same. Alright? It's kind of like having two different words and slight differences in spellings depending on, where you grow up, different regions of the U.S. Most people use English. Alright? But we all say things a little differently, we all spell things a little differently, and they're acceptable. Okay? Now, when you're going from this RNA nucleotide sequence, and you're rewriting it into a sequence of amino acids, this is the process of translation. This process of translation is basically like taking your notes in English and translating them into, let's say, Spanish. Alright, so a completely different language. Alright, so again, to reiterate, alright, the location for which transcription takes place. Prokaryotic cells, it's the cytoplasm. Alright, for eukaryotic cells, it's the nucleus. Alright, so translation. Translation occurs in the cytoplasm in prokaryotic eukaryotic cells, specifically at ribosomes. Alright, so there are three main players that are involved in this process of particularly transcription and mostly translation. Alright, so messenger RNA is a product of transcription. Alright, translation is going to involve the interplay of basically the ribosome, messenger RNA, and something called transfer RNA. And we'll talk about what each of these particular components do in the process of transcription translation specifically. Alright, so looking at the genetic code. Alright, so how exactly is it that this information in the DNA gets encoded into a protein? Alright, so think about it. You have 20 naturally occurring amino acids. And you have 4 DNA nucleotides. You have 4 DNA nucleotide bases. So how is it that these bases relate to, correlate to, correspond to a particular amino acid? How do you go from point A to point B? Well, you have 4 nucleotides in DNA and 20 naturally occurring amino acids. Alright, so here's the first scenario. What if one nucleotide corresponded to one amino acid? Well, for instance, alright, adenine would encode for the amino acid alanine. Alright, so in this case, given that example, 4 nucleotides, if it was a 1-to-1 relationship, 4 nucleotides would encode for only 4 amino acids. Well, that's not going to work because you have 20 naturally occurring amino acids, and we know that proteins are made up of more than just 4 types of amino acids. Alright, so that makes the first scenario obsolete. That doesn't work. Alright, so what about the second scenario? What if you have in sequence, you have 2 nucleotides that correspond to 1 amino acid? Alright, well, let's say that you have the sequence of guanine and thymine. And let's say that corresponded to the amino acid alanine. Let's say you have the combination of adenine and guanine. That would correspond to the amino acid histidine. Alright, well, in this case, you would have 16 possible combinations of nucleotides. In this case, you would only encode for a total of 16 amino acids. And we know that there's still more amino acids available. We know we have 20 naturally occurring. So this still doesn't work. Alright, so we're left for a third scenario. What if you had a sequence of 3 nucleotides that encoded for one amino acid? Alright, so for instance, ACC would encode for tryptophan, TGA would encode for threonine. Okay, well, based on the combination of 3 different nucleotides, or 3 nucleotides done to be different, you would end up with 64 possible combinations of triplet nucleotide codes. And you would encode for an overabundance of the amino acids. In this case, you would encode for all 20 amino acids. Alright, so this scenario, this works. Alright, so this triplet code, the sequence of nucleotides, the sequence of nucleotides ultimately in the DNA encodes for a specific amino acid. Alright, that triplet code is referred to as a codon. Alright, so you basically break up this sequence of nucleotides into triplets. Alright, they're not overlapping. Alright, so you break it up into groups of 3. Alright, and those groups of 3 nucleotides correspond to one particular amino acid. Alright, so here on the top you have your template strand. Alright, your template strand is transcribed into messenger RNA. So adenine is complementary to uracil, cytosine is complementary to guanine, cytosine is complementary to guanine, adenine is complementary to uracil, guanine is complementary to cytosine, thymine is complementary to adenine. Okay? And then this triplet code in the messenger RNA, this codon, corresponds to this specific amino acid, tryptophan. Alright, so you have to break it up to a triplet code. Alright, the second one codes for a different amino acid, the third for another, and the fourth for another. Alright, so here you're given a sequence of a template strand of DNA. Alright. The corresponding complementary sequence of the other strand of DNA is given here. Alright. So it's important to note the difference in polarity of the 2 strands. Alright. And the 2 strands are complementary. Here, you're given the same template strand, but you're going from DNA to RNA. Alright, again, note the polarity of the strands. They're anti-parallel, and the bases are complementary, but with the exception that where you would actually have a thymine is actually replaced with a uracil in the RNA. Now, this is something that you should be able to do. You should be able to give me the complementary strand of DNA if I give you a sequence of DNA nucleotides, and you should be able to give me a sequence of complementary messenger RNA transcript if I give you a template strand of DNA. Alright, this is something you should be able to do. Alright, so the genetic code. Alright, this controls how your codons are going to be translated into amino acids. Alright, so 3 nucleotides, or groups of nucleotides, correspond to a single amino acid. Now, again, you have 64 possible combinations, right? Well, there's 64 combinations. Out of the 64, you have 61 codons that correspond to amino acids. Alright, the sequence of nucleotides, it corresponds to the start sequence for transcription, or specifically translation, I should say, is AUG, and encodes for methionine. Alright, so this is a start site for transcription. Alright, so this is the beginning of a gene. You have 3 sequences of nucleotides. Alright, UAA, UAG, and UGA that are stop codons. Alright, they signal the stopping of translation. Now, these letters of genetic code in the DNA, they have to be read in the proper order, right, to make sense. Alright, so it's kind of like reading words in a sentence. Alright, so those letters that make up that word have to be grouped together so that they actually make up a word that has meaning. Alright? And these words have to be strung together so that they make a complete sentence. Alright? AKA a protein. A functional protein. Alright, so the sequence of nucleotide bases, alright, is referred to as a reading frame in the DNA. Alright, so a correct reading frame, for example, would be the red dog at the, instead of bug, the red dog at the, alright, bus. Alright. Actually, should be at the, it should be, excuse me, the red dog, not at, right, but ate. Yeah, I'll scratch that out. Ate the bug. That makes more sense. Alright. And the incorrect reading frame. Alright, so what if, for instance, you scratched out this T, didn't worry about it. Alright, and then you broke it up into a triplet code. Alright, you would get the her, ed, oga, tet, he, bug. Alright, that doesn't make any sense, right? Alright, so this sequence of nucleotides has to be read in the right order to make sense, to make a functional protein. Alright, so the genetic code. The genetic code is redundant. Alright, so in this case, you have more than one codon that encodes for a single amino acid. In this case, you have many different codons, many different sequences of nucleotides that encode for the same amino acid. Alright, that's a good thing, because let's say that you have a mutation. If you have a mutation in the genetic code, it's quite possible that mutation will result in the same amino acid being encoded for. Alright, so this is something that we call If that happens, and we'll get to mutations later on, that's something that we call a silent mutation. Alright, so a silent mutation kind of goes amiss. You don't see it. You don't see it unless you're looking for it, let me put it that way. Alright, because you have the same amino acid that gets encoded for, but there could still be an alteration in the genetic code. Alright? The genetic code is unambiguous. Alright, so what that means is that one sequence of nucleotides, one codon, encodes for only one amino acid. So you know that that sequence encodes for that amino acid, and that amino acid alone. Alright, there's no confusion. The genetic code is universal, quote unquote. Alright, for all intents and purposes, it's universal. Alright, there are some slight differences, alright, but we're doing some hand waving. Okay, so basically, the genetic code is shared between all living organisms. Alright, this is very useful, alright, when it comes to genetic engineering. And then the genetic code is read without punctuations. There are no commas, there are no semicolons, there are no colons, there are no periods, there are no question marks. Alright, it is read continuously. There are no gaps. Alright, it is one big run-on sentence with any kind of punctuation. Alright, so here, this is showing you how the sequence of messenger RNA, alright, so the letters, the codons, alright, so for instance, U, alright, that codon corresponds to the amino acid phenylalanine, alright, the codon CUA corresponds to leucine. Alright, notice that you have more than one codon sequence that encodes for the same amino acid. You have UUU and UUC that encodes for phenylalanine. Alright, so it's redundant. You have CUU, CUC, CUA, CUG that encode for leucine. Alright, so again, redundancy. Alright, so basically, this last little base, that last little base in the codon sequence, alright, it can change. Most of the time, it can change, and you end up with the same amino acid being encoded for. Alright, so it's kind of the floppy base. Okay, so another thing you see here is that CUU encodes for leucine, but CCU encodes for proline. Alright, so again, the genetic code is redundant, but it's also unambiguous because this sequence of nucleotides encodes for this amino acid. This sequence of nucleotides encodes for this amino acid in terms of the codons. Alright, so again, DNA is transcribed into messenger RNA. Alright, your template strand. Alright, so one gene can be found on one strand of parental DNA, and then another gene can be found on the other strand of the DNA. Alright, so particular genes are found on one strand or the other. Alright, so it isn't that one strand's always a template. It depends on the gene. Alright, so a gene in the DNA is transcribed to the RNA, RNA is translated into proteins.
The first step in protein synthesis is transcription of RNA from DNA. The portion of the DNA that is transcribed into an RNA molecule is called a transcription unit. An enzyme called RNA polymerase carries out transcription. With the help of protein transcription factors, it attaches to the beginning of a region of the DNA called the promoter, pries the DNA strands apart, and untwists a short portion of the double helix. RNA polymerase moves along the DNA, pairing up RNA nucleotides with their DNA complements, adding nucleotides to the end of the growing RNA molecule. Here's a close-up view of elongation. Only one DNA strand, the template strand, serves as a template for RNA synthesis. RNA polymerase moves along the DNA strand in the 3' to 5' direction, adding nucleotides to the 3' end of the RNA chain. Note that U in RNA pairs with A in DNA.
Once transcription is complete, RNA polymerase releases the completed RNA and detaches from the DNA.
Alright, so very similar to DNA replication. Alright. In DNA replication, you were synthesizing new strands of DNA nucleotides using an enzyme called DNA polymerase. In transcription, you're using the DNA as a template, and either strand of DNA can be a template for transcription. It just depends on where the gene is located and its orientation. Now, in this case, the DNA serves as a template for transcription. In this case, you have RNA polymerase playing the role of the enzyme that's going to synthesize RNA nucleotides. Now, one of the key things to note here is that RNA polymerase, unlike DNA polymerase, needs, or doesn't need, an RNA primer. Alright, recall that DNA polymerase required a pre-existing primer to add DNA nucleotides onto. RNA polymerase does not require this primer. Alright, so this involves complementary base pairing between the DNA template strand and the RNA nucleotides. Now, recall that RNA, you have uracil in place of thymine. Alright, so when you're looking at a gene in the DNA, alright, where exactly does RNA polymerase bind? How does it know where to bind? Well, there's a region that's upstream of a gene. Alright, so it's ahead of a gene, and it's referred to as the promoter. It promotes the movement and transcription of a gene. And at the end of the gene, you have a sequence of nucleotides that signal the end of a transcript. Alright, now the stretch of the DNA that's actually transcribed is referred to as a transcription unit. Alright, so there are 3 stages of transcription. Alright, so you have the initiation stage, you have the elongation stage, and you have the termination stage. Alright, so during initiation, alright, this is when RNA polymerase attaches to the promoter. Alright, it's going to move along the promoter, and then it's going to begin transcribing the beginning of a respective gene at the beginning of the reading frame of that gene. Alright, during this phase, you have RNA polymerase is going to add RNA nucleotides that are complementary to the DNA template, and as this RNA nucleotide is being assembled, it's going to move away from and out from the open bubble, transcription bubble, if you want to call it, and eventually RNA polymerase is going to hit a sequence of nucleotides and that transcription unit is referred to as the terminator sequence. Alright, it's going to tell RNA polymerase that it's reached the end of a gene. And now, RNA molecule detaches from the DNA template. Alright, so this is kind of walking you through the overall steps of transcription. Alright, so here, alright, so you have your beginning of your gene here. Alright, notice that upstream of the gene, you have your promoter region. Alright, downstream you have your terminator region. Okay. RNA polymerase binds to the promoter, and it's going to move along the promoter until it hits the reading frame. When it hits the reading frame, this is when it's going to begin to transcribe the DNA sequence into an RNA transcript. And it's going to move along the DNA, and it's going to move along the DNA in the 3 to 5 prime direction. RNA polymerase moves in the 3 to 5 prime direction along the DNA. The messenger RNA transcript is synthesized in the 5 to 3 prime to 3 prime direction. Alright, it's very similar to DNA replication. RNA nucleotides are always added 5 prime to 3 prime. Alright, until RNA polymerase reaches the terminator sequence. Alright, RNA polymerase falls off the DNA, and your RNA transcript is released. Alright, so during initiation, alright, so look at the promoter region of a gene. Alright, there is what's referred to as a TATA box. Alright, the TATA box has a sequence of TA repeats. Now these TA repeats, think about it. Think about the type of hydrogen bonds that hold together adenine and thymine. Well, it's double bonds. Alright, so these double hydrogen bonds are weaker than GC hydrogen bonds. Alright, because you have a triplet hydrogen bond for guanine and cytosine, right? Well, in this case, your transcription factors, alright, so there are TS or transcription factors that bind to this TATA box, and other components of the promoter that basically open up the promoter region so that your RNA polymerase enzyme can attach to the promoter. And it can begin to move along the promoter and begin synthesizing transcript as it hits the reading frame of that particular gene. Alright, so here you have RNA polymerase, it binds to the promoter. At the very end you have a terminator sequence. So during initiation, RNA polymerase binds to the promoter, it moves along the promoter until it initiates the initial transcription of the gene. Now, during the elongation phase, this is where RNA polymerase is moving along the sequence of nucleotides that make up a respective gene. Alright, and so again, it's always going to be in the 5 prime to 3 prime direction that the messenger RNA is assembled. But the DNA strand that's being read and the RNA polymerase moves along is in the 3 prime to 5 prime direction. Alright, so here during elongation, the segment of the gene is going to have RNA nucleotides that are attached until that RNA polymerase reaches the terminator sequence. And as the messenger RNA transcript is assembled, notice that it's kind of expanding out from the DNA. Eventually, RNA polymerase runs into the terminator region, where RNA polymerase falls off of the DNA, and the two strands of DNA reanneal. Alright, so it closes that open bubble, transcription bubble, and the messenger RNA is released. Alright, so messenger RNA. Alright, so we've already talked about the messenger RNA. You can split it up into a sequence of 3 RNA nucleotides, make a codon. That codon corresponds to a particular amino acid. So basically the messenger RNA basically gives you the sequence of amino acids. Alright, so messenger RNA in a eukaryotic cell has to be post-transcriptionally modified. It has to be edited, alright, because eukaryotic messenger RNA has introns and exons. Alright, so introns are basically non-coding regions between coding regions that we call exons. Alright, so what has to happen is that the messenger RNA has to undergo splicing. So in this case, RNA splicing involves removing the non-coding regions of the transcript, of the primary transcript, and leaving the coding regions, the exons, and basically annealing and joining those exons together. Along with that, you have a 5' guanine nucleotide cap that's added, and you have a 3' poly A tail. Alright, these modifications, the cap and the tail, alright, so they facilitate the movement of the transcript from the nucleus out towards the cytoplasm and towards the ribosome. It protects the messenger RNA transcript from cellular digestion in the cytoplasm, and it also helps the ribosome subunits attach to the messenger RNA transcript during the initial parts of translation. Alright, so here we're looking at RNA processing. Alright, so you have gene A, for instance. Well, gene A is comprised of 3 exons, so 3 coding regions, and has 2 non-coding regions that we call introns. Alright, so in this case, you have your pre-messenger RNA that's going to have a 5' cap attached to it. It's going to have the introns removed by the spliceosome, and then a poly A tail is added to the 3' end of the mRNA transcript. So this will be the mature mRNA. Okay, this up here is the pre-RNA, pre-mRNA, or the primary transcript. Alright, so once it's modified, it's ready for export into the cytosol. Alright, so here you have the spliceosome. Alright, so you're shown an immature RNA transcript with exons and introns. You have these spliceosome proteins that involve snRNA and other types of proteins that make up the spliceosome. You also have ribozymes that come in and aid in this process of editing the immature RNA transcript. And your spliceosome basically binds to the regions of the intron, it cuts them off, and you're left with your exons here that have to be glued together, and your introns released, and it gets degraded. Alright, so here you have a gene that has 3 exons and 2 introns. Alright, it goes through a process of transcription, RNA processing, and translation. Alright, so exon 1 corresponds to domain 1 of that particular protein. Okay. Domain 2 of the protein corresponds to exon 2, and domain 3 of the protein corresponds to exon 3. And this makes up the entire polypeptide, the entire protein. Alright, so basically you have regions of that protein that are encoded by different segments of the gene, different segments of exons. Alright, so when you're talking about transcription, you're comparing prokaryotic cells to eukaryotic cells. Alright, again, prokaryotic cells. Prokaryotic cells carry out transcription in the cytosol. Alright, there is no nuclear membrane. Alright, so that means that as the messenger RNA is being transcribed from the DNA template, what you can actually have take place is translation of that messenger RNA. Alright, because also the fact that this is all occurring out in the cytosol, alright, the ribosomes are right there, so they can begin to interact with the messenger RNA. You have your tRNAs, and all these things come together to aid in the synthesis of your polypeptide chain. Now also, the messenger RNA transcript produced by RNA polymerase, it doesn't have any non-coding regions. Alright, there are very, very few, if not any, introns in the genome of bacterium. Alright, bacteria organize, or their genomes organized into operons. Okay, so you have similar genes that correspond to a particular pathway that are transcribed together. Also note that here, that the ribosome is moving along your mRNA transcript, 5' to the 3' direction, and that your polypeptide chain is being assembled. Now, here you have an example of a polyribosome. You can have many ribosomes that attach to your messenger RNA, and you can have many proteins that are synthesized from one transcript. Alright, so here, look at this prokaryotic messenger RNA. You have your untranslated region ahead of a gene. Alright, now you have protein 1, alright, encoded by that particular region of the mRNA. You have protein 2, and you have protein 3. Alright, so from this one transcript, you have 3 proteins that are produced. Compare that to the eukaryotic mRNA. Eukaryotic mRNA, alright, is synthesized in a nucleus. It has to undergo post-transcriptional editing. Alright, so splicing out introns and gluing together exons. It has to have the 5' cap and the 3' poly A tail attached to it before it can even leave the nucleus. And then that one transcript will then be translated into a protein. Now here you have your mature messenger RNA. Alright, you have only one protein that's being produced from that transcript. As compared to prokaryotic mRNA, you have a series of genes grouped together in a genome that form an operon. And that corresponds to a series of proteins that are produced that are involved in a specific pathway.